Hello everyone, welcome back to another class on regeneration biology. Today we will learn about nerve dependent regeneration in axolotl. So in the previous class we saw how NAG contributes to the formation of the proper limb even if the nerve is removed. So we will see a more detailed view of nerve dependent regeneration in axolotl model. So the molecular basis of nerve dependent limb regeneration in an adult vertebrate, it's quite interesting. So the regeneration occurs only if there is simultaneous regeneration of the severed nerve. Means when you cut the limb, you also cut the nerve, and nerve also grows naturally through the regeneration, but the nerve also secretes the signal that is not just for nerve regeneration but for the whole limb regeneration. We usually think of nervous system as a carrying information, as a means of carrying the information between the nerve cells and their sensory and motor targets. Nerves are also essential for tissue regeneration because they are not just meant for their own regeneration but also for the tissue regeneration. When a salamander limb is amputated at any position, from the shoulder to the fingertips, you can start from here, and you can go up to here, fingertips. The stump forms a blastema. Whichever you cut, even you cut here, the stump or the remaining portion of the organ forms a blastema, a mound of stem cells from which is formed in that stump, from which the regeneration kickstarts or just begins. So if the nerves are cut at the base of the limb, deeper than the regenerative tissue, note it, the nerves are cut at the base of the limb, deeper than the regenerative tissue. That means here is the cut, nerve is cut from here inside. Okay, here is the cut. So that is what you should know. The limb stump is permanently denervated. The axons cannot regenerate. The limb regeneration fails because you have cut only nerve from inside. Limb is cut from here. So this nerve will not grow up to here and allow the growth of the limb doesn't happen. So after amputation, the severed axons retract. It shrinks. If the axon is cut here, it becomes short. It retracts within the stump and then grow back along the nerve sheath. First, if it is cut here, nerve is also cut, but the nerve now shrink back and then it bounce back. And you can see in the coming figure, but as of now we are just showing the message. The Schwann cells that make up the nerve sheath in this region that express the so-called NAG. NAG is from the Schwann cells of the Newt. Okay. The Newt anterior gradient protein, it's called NAG. As the first blastomal cells start dividing, later what happens, the NAG appears within gland cells in the specialized wound epidermis at the end of the limb. This epidermis is critical for sustaining later cell proliferation in the blastoma. So the initiation and the perpetuation is what is important. So the appearance of NAG in both the Schwann cells and the wound epidermis is abrogated by denervation. If nerve is removed, NAG cannot be formed because Schwann cells will not stay independent of the nerve. Hence, the Schwann cells will not secrete NAG. Hence, you will not get the proper limb formation. Isolated cells cultured from the blastoma, NAG promotes the proliferation in the denervated blastoma treated with NAG, and the wound epidermis is re-established. As you can see here, it's a Newt, limb is cut, and you can have the normal NAG protein that has been cultured. And normal situation, what happens is the amputated limb, you have the nerve, the black color nerve, it shrinks back and then it bounces back, and then it is allowing the blastoma formation that can start having the NAG expression and the limb is formed. But if the retracted nerve do not grow back, means the nerve is amputated much upstream. Denervation has happened. That is why nerve is not there. Then if you supplement NAG, you can end up getting the regenerated limb. So this NAG protein can be made from a NAG producing cells even in culture. So regeneration of salamander limbs requires concomitant regeneration of the severed nerves. That is, a single protein NAG can substitute for regenerating nerve cells. So NAG is one of the early molecule that a wound epidermis is expecting to be released only by the Schwann cells. But this doesn't mean that NAG is good enough. NAG is good enough to complement the absence of the severed nerve, or to compensate for or supplement for the loss of nerve. Neuroglin signaling is essential for nerve-dependent axolotl limb regeneration. Those who are interested can read this article. The Mexican axolotl, Ambystoma mexicanum, is capable of fully regenerating amputated limbs. Similar to NAG, axolotl also use a molecule called neuroglin. So the molecular basis behind this phenomenon of limb regeneration remains poorly understood. You know a lot, but you do not know as much as to create a limb in a mammal. But the previous studies have suggested that nerves support regeneration via the secretion of essential growth promoting factors. A neuronally secreted proteins known as neuroglin, in short form it is called as NRG1, fulfills all these criteria in the axolotl. Immunohistochemistry and in situ hybridization of NRG1 and its active receptor ERBB2, that is the receptor, revealed that they are expressed in regenerating blastema but lost upon denervation. You may remember that when we studied about epidermal growth factors, we discussed about ERBB receptors. Different receptors are there in the previous class. We discussed ERBB 1, 2, 3, 4 like that, and how they form homodimerase and heterodimers based on the ligand. That time also we discussed about the neuroglin, but that was regeneration of some other tissue, say intestinal epithelium, etc. But here we are talking about limb regeneration. So NRG1 was localized to the wound epithelium prior to blastema formation, and it was later strongly expressed in the proliferating blastemal cells. Supplementation of or the implantation of NRG soaked beads rescued regeneration to digits in the denervated limb, similar to that of NAG in the Newt. You can see in axolotl also the NRG1 contribute, or it can supplement the absence of nerve connection to the wound site. Pharmacological inhibition of NRG1 signaling reduced cell proliferation, blocked blastema formation, and induced aberrant collagen deposition, that is nothing but scar formation, in fully innervated limbs. So innervation is there, you did not denervate it, innervation is there, amputation is there, but you blocked NRG1. So nerve may be secreting some other factors also, but absence of NRG1 is powerful enough not to have proper limb formed. That is indicated by aberrant collagen deposition. The nerve-dependent NRG1 ERBB2 is the receptor, ERBB2 signaling promotes blastemal proliferation in the regenerating limb and may play an essential role in blastema formation, thus providing insight into the long-standing question of why nerves are required for axolotl limb regeneration. We knew nerves are needed, but we did not know why nerves are needed, and these experiments clarified that. NRG1 and ERBB2 are expressed in the PNS and the regenerating blastema. So how people explored it, so we will get into the different details of this experiment. So this is NRG1 in situ and of all types, and here NRG1 type 1 and type 2, and also you can see different location in the blastema. This is a growing blastema, And you know, it is an in situ hybridization. It stands for in situ hybridization. That means you are detecting the RNA of NRG1 isoforms and receptors are expressed in the blastema of 14-day post-amputation. Insets show sense control. Sense control means antisense is the usually used as a probe. Sense control means you are using the same gene as a probe. So same gene will not hybridize with the same gene. You need an antisense of that gene for hybridization. So sense control means there will not be any signal. And also RT-PCR analysis showed that the EGF like proteins, they are also induced, and NRG1 is induced, and also the receptor ERBB2 are induced, and they are being confirmed by statistical analysis. So in a nutshell, what you understand, both the ligand and the receptor are induced in the blastema, so that nerves, when secrete the NRG1, it will act on the ERBB2 receptor and will do the job what it is supposed to do. That is the blastema formation. NRG1 and ERBB2 are expressed, like I told you, in the peripheral nervous system. And this picture that shows how different phases like G to K panel, what you are seeing here is the NRG1 and ERBB2 are expressed in the mesenchyme of the regenerating blastema but lost upon denervation. So in normal scenario, if nerve is present, amputated severed nerve is present, NRG1 and ERBB2 receptor, everything happens smoothly. But if nerve is not there, it is denervated, then you will not have blastema formation at all. You will have aberrant deposition of the collagen. Green and orange fluorescence is due to autofluorescent cellular debris. Normally in tissues, you will get unwanted fluorescence based on what tissue you are studying, but that can be ignored. And L to O panel, what you are seeing is NRG1 and ERBB2 are expressed in the dorsal root ganglia and peripheral nerves. And this P and P dash, what you are seeing is NRG1 is expressed in the wound epithelium, that is WE, and the mesenchyme. Wound epithelium and the underneath is the mesenchyme cells of 6-day post-amputated limbs along with the proliferating BRDU positive cells. And in Q and Q dash, what you are seeing is extensive NRG1 expression and co-localization with the BRDU in 16 DPA blastema. Co-localization with the BRDU indicates NRG is expressed in the proliferating cells. Arrows indicate the co-labeled cells, both BRDU and the NRG1 expression. NRG1 and BRDU co-localization along with the peripheral nerves in the regenerating limb at 16 day. Earlier was initial like 6 DPA, now you are 16 DPA. Denervation significantly decreases the percentage of BRDU and NRG1 localization in the panel S at 16 DPA because you waited until 16 DPA but you still don't have the blastema formation because of the denervation. In panel T, the western blot of the NRG1 at 16 DPA, it shows a band at the expected size of 47 kilodalton and a greater band intensity in the blastemal tissue relative to the denervated tissue. So what you are seeing is in denervated tissue, you do not get a proper NRG1 expression. So what you learn from here is that these researchers, they show that NRG1 depends on the presence of a severed nerve. So supplementation with NRG1 rescues regeneration in denervated limbs. So you can see here the limbs are amputated and wound healing happens, kickstarts, and the beads are implanted, that is at the denervated limbs which are soaked with NRG1, and the blastema starts forming and limbs are reformed or they are formed again. They act as if they are re-innervated. That feeling comes because you soaked beads are exposed and the blastema formation and limb formation takes place. So panel A is the timeline of NRG supplementation experiment, and it's supplemented with NRG soaked beads up to, and this is, they have gone up to around 20-21 day post amputation. Arrows indicate the plane of amputation. In panel E, from 6 to 20 DPA, NRG1 supplemented, up to 7 individuals they have done it, and limbs regenerated significantly more tissues than denervated animal. As you can see here, innervation normal blastema, denervation you don't get blastema, And when you have denervation, when you don't have any supplementation with NRG1, you get only a stump. So it is somewhat closer to innervated scenario, but definitely drastically different from that of a denervated scenario, because denervated scenario you don't have any blastema that is formed. So NRG1 supplemented limbs are regenerated in the absence of innervation or in the presence of denervation. So supplementation with NRG can rescue the denervated limbs, and in this panel what you are seeing is the timeline of late NRG supplementation. Instead of giving early stage, if you give in a late stage, what happens? Implantation of NRG1 socked beads into the denervated limb rescues the regeneration into the point of digit formation at 36 dpa. Until the digit formation time you wait it can work. Arrows indicate the plane of amputation and the dotted lines outline the regenerating tissue, as you can see in this picture. So panel M, what you are seeing here, NRG1 supplemented limbs regenerated significantly more tissues than denervated, and it can lead to formation of the proper limb itself, and significantly less tissue than innervated. Means supplementation is not as good as a proper nerve being present, but it does the job. And N to O panel shows Elsian blue staining showing digit formation in control and NRG1 treated limbs. So NRG1 induced growth and digit formation in fully denervated limbs. That is the message we have to take. So inhibition of ERBB2. Now you are going into the receptor. So far we are playing with the ligand. ERBB2 receptor, if you block it, it blocks regeneration, inhibits proliferation, and induces aberrant collagen deposition. Means as good as blocking or as good as denervated scenario, even though nerve is present. So A to C panel, inhibition of ERBB2 with the 500 nanomole mubritinib blocks the blastema formation. It's a drug at 13 dpa. The picture is taken at 13 dpa because that is a time when you can have a proper limb bud that is being formed, as you can see in the innervated scenario. When you treat with mubritinib, it is as good as a denervated scenario because the stump is not growing, but whereas the stump from here, it has grown up to here. And D16, this is D0 treatment, and D16 also you can see, you are able to see a proper limb not formed as good as denervated scenario, whereas in the innervated scenario, it started forming the digits. So D to F, the mubritinib application after 16 dpa, much later, blocks limb proliferation but not patterning, and appears phenotypically similar to 16-day denervation. 16-day denervation, what you get it, an aberrant limb that will be formed. It looks like that even if you blocked much later stage. Dotted lines outline the regenerating tissue. Blocking means in this whole panel, you are blocking ERBB2 receptor only. Picross Series staining showing at 23 days of submersion in 10 micromolar, little higher concentration. Earlier they have done is 500 nanomolar. Now you are doing higher concentration. 10 micromolar mubritinib results in the contraction of the epidermis. Means very severe blocking of ERBB2 receptor takes place, and an aberrant collagen deposition is seen. In the mesenchyme, the contrast to the minimal fibrotic deposition is seen in controlled blastema. So you can see a huge contrast compared to a 10 micromolar mubritinib treated limbs. And the dotted lines indicates the boundary of the epidermis. So now to continue further, the Masson's trichome staining of control and the mubritinib treated limbs at a 12-day post-amputation show a lack of blastemal accumulation in the drug treated limbs. Drug means ERBB2 is blocked. That is, you can see in I and J panel. K, L and N panel, treatment with the mubritinib does not reduce innervation but significantly decreases the proliferative index of the amputated limbs. Dotted lines indicate the plane of amputation in this picture also. At 14 dpa, M panel, 14 dpa, mubritinib treated limbs had regenerated significantly less area than the control. So, but not as low as denervated. That means presence of the nerve helps in some way, but not adequately enough to cause a regeneration if ERBB2 receptor is blocked. So that NRG neuroglin has to act on them. Panel O, limbs that were either denervated or treated with mibrutinib at 16 dPa regenerated significantly less tissue than the control limbs. That is what has been plotted here. You can see different innervated, denervated, and innervated plus mibrutinib. Arrows indicates the plane of amputation in there as well. So EGFR inhibition. Now we are talking about another receptor which belongs to the same family, epidermal growth factor receptor, because they also act on through this ERBB1, ERBB2, ERBB3, like that, different ERBB4. You can refer back our earlier class, we discussed about that. EGFR inhibition inhibits wound closure and is phenotypically distinct from ERBB2 inhibition. One may wonder, because of blocking of ERBB2 receptor, you blocked epidermal growth factor signaling, but no. It can specifically block neuroregulin function. That is what has been shown here. A to C panel, what you are seeing, proliferating cells are localized to the mesenchyme in the AG1478 treated limbs, that is a EGFR blocker, and the epidermis in the mibrutinib treated limbs at 6 dPa. So two experiments, one EGFR is blocked, in another ERBB2 receptor is blocked. So EGFR is meant for all EGF signaling, whereas that is this drug AG1478, whereas mibrutinib blocks ERBB2. So keep that in mind. And the arrowheads indicates autofluorescent cells. You can ignore them, debris and rod cells. Dotted lines indicate the boundary between the wound epidermis and the mesenchyme. So keep in mind, in the beta tubulin 3 and BRDU and DAPI staining is been shown here, and you can see the proliferating cells are localized in the mesenchyme. So although the nuclei is present, and the BRDU, the green color BRDU indicates the actively proliferating cell, but when you have EGF blocked, you do not see too much of the proliferation that is occurring in the blastema cells. Whereas in the same way, in the mibrutinib treatment also, you see a significant decline. There is some stump of cells that are being formed. They do not properly form the proliferative blastema, even though the nerve is present. So what you learn from here, that ERBB2 receptor blockade affected the blastema formation in spite of having the neuron, is not through EGF signaling, but through NRG1 signaling, neuroregulin signaling. So EGFR inhibition inhibits wound closure and is phenotypically distinct from ERBB2 inhibition. This is another evidence. It can EGFR needed for EGF signaling on EGFR receptor is needed for wound closure to happen. So if you block EGF receptor, wound closure get affected. That is what has been shown here. So AG1478 is for blocking the EGFR, and this is the control. You can see here in both conditions, 3 dPa, 7 dPa, and also H and I is a later time point, as you can see here. H is for 10 days, much later, 10 days. So what we see is AG1478 treated limb showing aberrant wound closure over the time compared with control limb of 3 dPa and 7 dPa. Both scenario, that is what you are seeing. In H, in panel H, what you are seeing is here, limb treated with AG1478 for 10 days demonstrating aberrant development of iridiophores, which is needed for the proper melanin and the pigmentation, etc. that has to happen, the skin pigmentation. I panel is the control limb treated with DMSO for 10 days showing lack of iridophores. So there is an aberrant migration of iridophores are seen in EGF signaling blocked scenario. Regeneration is compromised even there also. So why EGFR signaling is been selectively shown here? Even if NRG signaling is normally occurring, absence of EGFR also can interfere. So that's why I have told in many classes, absence of one signaling, the regeneration is blocked. That doesn't mean that that signaling is the one and only signaling that is contributing. Other signaling is also, just like if you are cooking, if you just forgot salt, it will not be a tasty food at all. But you can claim, oh, I put only one rest, all has been put, but it will not be a tasty food. So every component has its own role. So percentage of proliferating epidermal and mesenchymal cells are compared in control in the J and K panel. Mubretinib treated and AG1478 treated limbs at a 6-day post amputation. It is done in around 5 to 6 biological replicates. So if you look closely, NRG signaling, what do we learn from NRG1 signaling? A single nerve-derived protein known as neuregulin-1 is capable of supporting blastomal growth and tissue regeneration up to the point of digit formation in the denervated axolotl limb. So keep in mind, neuregulin signaling, if it is blocked until certain stage of development, if you could supplement the neuregulin before the digit formation, you can still rescue the regeneration. So the system is so plastic and it is wanting to regenerate. So that is the idea you should gain from these studies, that regeneration is very much normalcy in this animal, whereas absence of some molecule such as neuregulin can completely shut it off. So it would be interesting to look why these genes are not running on in mammals, or can we learn a lot from them, etc. Nerve dependent NRG1 ERBB2 signaling is crucial for blastomal proliferation and may also be an essential component of blastoma formation and scar prevention. They are scarless regeneration, scar-free regeneration programs. Scar also is a bottleneck in the regeneration program. NRG1 is the first protein known that has been shown to be capable of rescuing regeneration to digits, up to the digits in an axolotl limb. Newt anterior gradient protein NAG has also been shown to rescue regeneration in the denervated Newt limbs. It may be interesting to see, can NAG do the job in a axolotl, or can NRG1 can do a similar job in Newt? So such experiments also will tell how plastic or how the system can talk to each other. Exploration of relationship between these two signaling pathways is necessary in order to characterize fully the underlying cause of nerve dependency in axolotl limb. We know nerve is needed. Now we know more why nerve is needed in Newt as well as in axolotl. Given the conserved role, conserved means evolutionary conservation across a diverse species, NRG1 and ERBB2 signaling in the peripheral nerves, as well as its necessity in other animal models of cardiac and peripheral nerve regeneration, elucidating the function and mechanism of this signaling pathway in axolotl. This pathway may be may have far-reaching impacts on the field of regenerative medicine. So we should understand, or we should try to explore, the NRG1 is contributing at what level, until what time it is continuing to produce. Is it produced till the last digit is being formed, or is the level of NRG is changing, or does this formed NRG still actively doing the ERBB2 receptor binding? All these things one has to look into, because it is very much possible the NRG released in the very beginning may not be released at the same level till the fag end of regeneration. So these are all the fine-tuning observations and fine-tuning analysis one has to perform in order to fully understand the limb regeneration. We will learn more about limb regeneration in the next class. Thank you.